So, hello everybody, and welcome to the ESDR Kitchen. I'm Eli Sprecher from the Department of Dermatology at the Tel Aviv Medical Center in Israel and an ESDR board member. And on behalf of the board and my co-chair, Professor Bernhard Homey, I'd like to welcome you all for this new episode of the ESDR Kitchen. I'd like to remind you that the ESDR Kitchen program, as well as all the previous episodes, are available on the ESDR websites. We are all invited to look at those. Today, we are particularly honored to welcome Professor Martin Röcken for a molecular cuisine episode. So in short, about Martin's long and amazingly successful career. Martin Röcken is Professor of Dermatology and Chairman of the Department of Dermatology at the University of Tübingen. He's not only a world-renowned scientist, he has and continues to contribute significantly to the dermatology community. He's been playing numerous functions within our society, was treasurer and president of the SDR. He is an LDS board member and he's chair of the scientific program committee for the World Congress of Dermatology, which is due to take place in Singapore in 2023. And he is president-elect of the EADV. His laboratory has been exploring the capacity of adaptive immunity to prevent or help treating a wide range of pathologies, including inflammatory conditions, infectious diseases, and cancer. He's especially interested in this context in understanding the signal by which T cells can impair or promote cancer development, including melanoma and inflammation within the skin. And in fact, his research has brought about the radical changes in central concepts in dermatology over the years, and he will review many of those during his talk entitled Psoriasis from T cell exclusion to cytokine-based therapies. I'd like just to mention that this episode is supported by Pfizer. Remind you all that you can forward your queries to the chat or the Q&A functions. And that's it. Martin, thank you very, very much on behalf of all of us for your willingness to speak today. Please.
Oh, I will start with psoriasis from T cell exclusion to cytokine. We all are used now for many years to use cytokine-based therapies, but it started. But can I continue now? Okay, this way. But it started differently. In the beginning, in the 80s, dermatology was quite descriptive from the clinical and histological part, and there was a big discussion whether it's a primary disease of the keratinocytes or it's a primary disease of the immune system. And in the 70s and 80s, cyclosporine A was developed for patients who had grafts, and especially kidney grafts. And then it was observed that patients receiving cyclosporine A after kidney transplantation had a strong improvement of psoriasis. And in the 80s, the study started to investigate cyclosporine A in psoriasis. It was shown that it really, a molecule which impairs T cell activation, that it really improves psoriasis in many patients. And by histology, we see on the left the mononuclear cells that on the right side, upper side, are stained with CD3, and what shows that most of the cells are T cells. So this was one side. The other side is to develop novel therapies, we have to start always with first in vitro and then in vivo experiments. And what was or is still an important model to investigate the possibility of an immune therapy is mice with an experimental allergic encephalitis. It was abbreviated EAE. And it was shown that the disease can be induced, I show later, by the transfusion of autoreactive T cells. And on the right side, we see a healthy mouse, which I show later where it comes from. And on the left side, a mouse with multiple sclerosis-like disease with a paralysis of the tail and the legs. And this model has been used to develop many novel therapies. And we have three different approaches. One approach, which we are using now, is a modification of the cytokine pattern of the infiltrating T cells and the antigen-presenting cells. But there were different approaches which were first-line therapies or thought to be more important than cytokines, or even one didn't think of cytokine therapy. This was a deletion of T cells with toxins, which comes from the late 80s, early 90s. And at the same time, the other approach was that one discovered molecules that are important for the immigration of T cells into tissues. And they were started to block the development of autoimmune diseases. So the first approach was to inhibit the T cell migration into the target organ using, again, EAE. And there are different approaches. One was to block the emigration by inhibiting adhesion molecules or by inhibiting integrins to impair the interaction between the immune cells and the basement membrane or the adjacent cells. And this first paper on this was published in 1992 in Nature to inhibit EAE in rats. And this was the pre-drug for natalizumab, which is now used in multiple sclerosis. And a similar drug, which also blocks integrin interactions, velodizumab, who is now used to treat inflammatory bowel disease. Prior to natalizumab and velodizumab, it was used efgartigimod and alifacet to inhibit the migration into the skin. The efgartigimod was arrested. It was not so efficient, had about 20 to 25 patients, but in rare cases, it caused PML, leukoencephalopathy, virally induced, and that's why these drugs were stopped. And alifacet was only approved in the US, but not in Europe. But this principle, I can't continue on this, is currently pursued in cutaneous T-cell lymphomas. The second mode to exclude T cells to improve EAE was based on a famous Cell manuscript, which showed for the first time that mice that were injected with autoreactive T cells or were actively encephalitis was induced, cured the encephalitis, which is the severity score, where high severity score is 3, lots of paralysis, 1 is medium, by deleting with an anti-V beta chain antibody the pool of the autoreactive T cells. And since T cells are also found in psoriasis, and if we stain these T cells bear activation markers, especially they are loaded with the IL-2 receptor, the concept was to bind a toxin to an antibody or to cytokines that bind to activation molecules. And one of the most famous papers was by Jim Kruger, which was published in the late 90s. They showed that a toxin coupled to IL-2, and the IL-2 binds then to the activated T cells, really improved psoriasis in a number of patients. And this supported further the idea that psoriasis is caused or mediated by T cells. So the deletion of the T cells, well, proof of concept studies were done in psoriasis. And again, this is pursued as a concept in the treatment of lymphomas. We started with a different approach. What we observed together with Martin Röcken, who is a neurologist, who was a neurologist at NIH, that the T cells that cause the disease in the mice are mainly interferon gamma. At that time, we could not yet measure IL-17, and we now today Th17, IL-17 producing cells. And it was known that the Th1 cells that produce interferon gamma, what is green, are induced by IL-12. Today we know that IL-23, I'll show later, induce IL-17, and we knew that IL-4 induces T cells that produce large amounts of IL-4 and especially IL-13. And we had the experience from infectious diseases that interferon gamma producing T cells can use strong local inflammation and control and also kill parasites and other pathogens, while interleukin-4 producing T cells are not capable and rather promote the growth of the pathogens, and many animals are dying if they have an IL-4 or Th2 associated immune response to pathogens. So what we asked, as autoreactive cells can induce organ-specific autoimmune diseases, we asked whether it's possible not to delete the T cells, but to leave the autoreactive cells inside the animal or in the humans, and just to change the cytokine pattern from a pro-inflammatory Th1, now also Th17 phenotype, to an anti-inflammatory Th2 pattern. So we used again this mouse and analyzed the brains after inducing the EA, and we found a lot of interferon gamma, especially no IL-4. IL-17 was not investigated in the initial periods. And if we now go further, we had developed at the same time that the stimulation of a T cell in the presence of interleukin-4, always you need two signals, the T cell receptor signal and interleukin-4, is capable of inducing in an antigen-specific way Th2 cells. So the question was, if we have a running autoimmune disease, as the T cells, the autoreactive T cells are stimulated by brain proteins, is it possible to deviate the immune response from a Th17, Th1 response to Th2 response in the presence of autoreactive cells? And that's now the real experiment, what we have done. The mouse left, both mice received the autoreactive T cells, Th1, Th17, but the second mouse was vaccinated with interleukin-4. And what we did induce organ-specific, MBB-specific Th2 cells. And this was published with Michael Röcken in the Journal of Experimental Medicine. And as we can see, it was studied by an independent pathologist that the T cell infiltrate in the brain is similar in the upper and in the lower part. In the upper part, the destruction of the brain tissue comes from the T cells and the cytokines, especially probably TNF, while in the brain, lower brain part, what we see, the structure of the brain is almost normal, and the mice, as you saw, are running around normally. So this was then published as cytokine-induced immune deprivation as a therapy of autoimmune diseases. And when I returned back from the United States to Europe, it has to be the kitchen, a little bit of private story too, we asked whether we can translate this concept from mice to humans. And the disease we did select was psoriasis, because of psoriasis is, as we know so far, not associated with antibodies, and IL-4 producing T cells are strong stimulators of antibody production. So we used psoriasis, analyzed the T cells, and found that the T cells in psoriasis bear the marker CCR5, which indicates these are either interferon gamma or IL-17 producing T cells. And we have here again a picture of psoriasis with the infiltrating mononuclear cells. And the question was, can we deviate these Th1, Th17 cells into Th2 cells? So we did exactly the same as we did previous studies. To analyze the amount of IL-4 you need to deviate human Th1 cells into Th2 cells. And we speculated that these are autoreactive T cells stimulated in the psoriatic skin and added or treated the patient with IL-4. And these are the results. That's one of our best patients, of course, who really cleared within 4 weeks. And if we take the overall, the decrease in the PASI score within 6 weeks was really dramatic. But what we also see is that they responded to all drug concentration, but in a different manner. Those who are in the darker lines responded much better. These are those that are capable of inducing Th2 cells, while the clearer signs not. And what we observed is a clear induction of interleukin-4 within the skin. We also found in the skin, as a log scale, relatively mild reduction of the interferon signal. And we sincerely wrote in this paper that we had mainly induction of IL-4 producing cells, also in the peripheral blood, a minor change or even sometimes an increase in the blood of interferon gamma production. And we did not answer why IL-4 improved the psoriasis or EAE, because of also in EAE, we found that there was an increased interferon gamma production within the T cells. So there was somehow that IL-4 did suppress psoriasis, did suppress EAE, but it was not clear why. We published this, and we sincerely wrote that there are still lots of interferon-producing circulating T cells. So at the same time, pharmacology then started a little bit later, started to develop antibodies to block IL-12 with the goal to block interferon gamma. And the first antibody was ustekinumab. And it was thought that ustekinumab blocks Th1 development. It was solved this way by blocking interferon gamma. And this by Chris Griffiths, this was the first cytokine treatment targeted T cell cytokines worldwide by Chris Griffiths and Bruce Strober, who showed that ustekinumab blocks, improves psoriasis. You see, these are the first data on it, with showing that you get about 60% of patients who really improve. What we know is, what we asked is, interferon gamma really the correct cytokine to block? Because of, if we look to the genetics, especially the early work by J.T. Elder from Ann Arbor, who showed that there's little or no interferon gamma signal in psoriasis, but there's a strong signal for IL-12B, which co-localizes with IL-23A, and which works also on the suppression of IL-23. And we find IL-13, which is associated with psoriasis, with the IL-4 pathway and the TNF pathway. So future work showed that we find in all the lesions interferon gamma, but in the new lesions, we found CD4 T cells together with IL-17 producing T cells and IL-23 producing monocytes or antigen cells. And never we found any, and others neither, IL-4 within the psoriatic lesions. So at that time, it was early 2000 years, that one found on the one side abnormalities in the TNF family, and then IL-23, IL-23p19, the IL-23 receptor is also called, this is a specific one, and the IL-2012, IL-23p19 common receptor, and defects in the IL-4, IL-13 family. So we had to switch our gears and ask, is really ustekinumab working by blocking interferon, or is rather working, what is more logical, by blocking IL-23? So it's inhibition of the p19, p40 molecule, which then binds, as we see here down, to the IL-23 receptor. And it was then Kamran Qureshi, who is now the chair in Berlin, who went from our lab to John O'Shea's lab and used there the EAE model, and there he showed that the IL-23 educated Th17 cells are those who induce a severe EAE, with the clinical showed in the black dots, while Th17 cells that are not educated by IL-23 fail to do so. So only the IL-23 educated T cells then induced, he showed, the severe type of EAE. The question was, does this also hold up for psoriasis? So we went back to our studies and analyzed them for IL-23 before and after treatment, and for the presence of Th17 cells before and after treatment. And what was shown that before treatment, the skin is full of IL-17 producing T cells. The CD3 shows the T cells in blue and the 17 in red. And the MHC class 2 positive cells are full of IL-23, as shown by the red staining for IL-23. And if you check this by PCR, quantitative PCR, there is a dose-dependent decrease of the IL-23 mRNA between week 0 and week 6 of treatment in the patients, and those who had the best responses had the lowest IL-23 and lowest IL-17 mRNA. And these are the corresponding figures. We had sustaining for IL-17 and CD3 before, and now here after therapy, and the same for MHC class 2 positive cells and IL-23 and TC. After treatment, no IL-17 and IL-23. So this showed that the treatment improvement was not associated with the reduction in interferon gamma, as we already showed in the original paper, in the Nature Medicine paper, but was associated with a loss of IL-23 and a loss of IL-17. Suggestions that ustekinumab works mainly by blocking IL-23, but we have still an antibody binding to 2 signaling molecules. So the question then rose, also by many others, is it really IL-17 and IL-23? And these are now the clinical data. That's blocking IL-17 with ixekizumab, and it shows that only blocking IL-17, the effector cytokine really improves psoriasis by 90%. The same brodalumab, which binds to the IL-18 receptor, or the cytokine, which induces, if you block the cytokine that induces the Th17 cells, if you block IL-23p19, then we get also a strong reduction of the PASI score by 80% in most patients. So these were the proof of concept studies that really the IL-17 and the IL-23 producing T cells, or the IL-23 producing APC and the IL-17 producing T cells, are the main mediators for the initiation of the psoriasis. And this has been now translated to develop novel therapies for IBD, rheumatoid arthritis, multiple sclerosis. There was this drawback of IL-17 antibodies in the case of inflammatory bowel disease, which is not observed with anti-IL-23 antibodies, probably due to the fact that different types of IL-17 are blocked by either therapy. So what did we learn over the past 20 years? That CD4 T cells and neutrophils are main drivers for many organ-specific autoimmune diseases. That cytokines decide whether autoreactive T cells are harmful or protective, and that correcting aberrant cytokine production provides long-term protection from autoimmune diseases. But we also learned that overcorrection may promote cancer and see infectious diseases because of, we saw that ustekinumab improves psoriasis in about 60% of patients. And there was constructed an antibody against the same molecule with a high affinity, which almost blocked the entire IL-12 production. And the study and the approval of the drug was stopped shortly before they wanted to market it, because they had increased numbers of infectious diseases and even 1.4% of patients who developed during the therapy cancer. And this was published in the Journal of Investigative Dermatology. So overcorrection may promote cancer and severe infection disease by deep suppression of IL-4 and interferon gamma. So I want to thank you for your attention, and all colleagues of the department, all the former colleagues of the Department of Dermatology for their commitment in dermatology and in further development novel therapies and science in dermatology. Thank you.
Yeah, thank you very much, Martin, for this excellent talk and the fascinating journey. I want to encourage everybody in the audience to give us your questions via either the chat or the Q&A function. And since we do not have questions yet, I would like to start to ask you, Martin. So right now, with the treatment of dupilumab, so anti-IL-4 receptor alpha treatment, we see that there is a balance in the cytokine networks. And in some patients that are treated for atopic dermatitis, we all of a sudden see that they show typical psoriatic plaques. If we think about this balance you showed from IL-4, that you can shift Th17-driven inflammation and treat it, and we now treat even young kids at the age of 6 with dupilumab, would you anticipate that in an early immune system, we could also drive the immune response then more towards the Th17 inflammation? And do we have to have a look at diseases such as MS, EAE-like inflammation, and what could predict an individual that on the one hand has atopic features and then all of a sudden develops Th17 driven psoriatic or EAE-like inflammation?
With the EAE, it's more difficult to judge because of there's a dichotomy of IL-4 and the effects of IL-12 and IL-23. IL-4 promotes the production of interferon gamma by dendritic, but deeply suppresses IL-23. So I think for the diseases driven by Th17 cells, suppression of IL-4 may really wake up the dormant disease, perhaps psoriasis, in patients who have genetic predisposition for both. So I think that's a real risk. For multiple, they are too young, fortunately, the children, and I would be careful giving dupilumab to patients with multiple sclerosis.
Yeah, of course. Yeah.
I didn't never do. So what could you use? Probably the IL-13 blocking drug, because of the effect of IL-4 as a master regulator in inducing differentiation of antigen-presenting cells is much stronger than the effect of IL-13, which is more on the B cell side.
Yeah. We got one question that is dealing with the increase in cancer, whether this is also occurring to your knowledge with anti-IL-17 or anti-IL-23.
No, probably not. They seem to be relatively safe because of the cancer immune therapy seems to depend mainly on IL-12 and interferon gamma, and the role of IL-17 and IL-23 in cancer development is still entirely unclear. There are some data even that may suggest that they can promote it. So for the moment, it seems not to be a risk to give anti-IL-17, anti-IL-23 in cancer patients. Let's say we shouldn't give it to cancer patients, but there's no signal that it promotes cancer development, as far as I know.
Yeah. We got another question. Is there a mechanistic explanation for the presence of monoclonal antibodies resistant psoriatic plaques? This is the first part. So an explanation for treatment resistant plaques.
There are some patients. This was especially elaborated by Michel Gier and you too. I think you were involved in the studies that in some patients, psoriasis is mainly driven by type 1 interferons. And maybe that these types of psoriasis was shown by paradox reactions to TNF. Most are driven by Th17, but in some rare cases, they are driven by interferon. This may be the reason for some of the plaques that do not respond to anti-IL-17 and anti-IL-23. It's about 5% of the patients who have this type of different response causing psoriasis.
So, Eli, do you have any further questions? There are no more in the chat. And
I could just ask a quick question, but I'm not sure if the answer can be short. So when you look, Martin, with your perspective over all these years, when the field of psoriasis and inflammation has gone through so many changes. Now coming back to the keratinocytes and looking at their real function, especially in view of the clinical data that we are now collecting with small molecules that are targeting keratinocytes. So do you still see the pathogenesis of psoriasis in terms of this kind of dichotomous view, yes, where it is either that or that? Or do you
I think it's a very important point. If you look to psoriasis, to inflammatory bowel disease, to sometimes of fatty liver disease, to multiple, we find a similar way of inflammation. But we have different organs. And I think we need for both, for all the diseases, a specific susceptibility of the organ, a reaction type of the organ. And you don't induce with Th17 cells in the normal skin of a non-psoriatic predisposition person a psoriasis. So I think that the immune system is a good target for treatment, but probably the genetic background is in the keratinocytes or perhaps even the fibroblasts. We don't know. But I think the target organ is the organ who is really responsible for the disease manifestation, but the immune system triggers the disease, and on one side and on the other side, it's relatively easy to target it for therapeutic purposes at the current level, what we know.
Thank you.
Would you agree as a geneticist?
Absolutely.
So, Martin, this is a perfect concluding statement. Thank you very much for these excellent insights, and we are very grateful for the support of Pfizer, for you, for your attention, and I'm looking forward to further series of ESDR Kitchen and Molecular Cuisine. Have a great day, and thank you very much again for everybody. Bye-bye.
Thank you. Bye-bye. Have a good afternoon.
You too. Bye-bye.
Bye. Thank you.
Thank you.